Hi Year 12s, welcome back to another video. So we're just going to go over the recap really quickly. So we have our 3 stages of gene expression, transcription, which we looked at in a previous video, RNA processing, which we're going to touch upon in this video, and then translation, which will be in the next video. Okay, and that's essentially the process of DNA being turned into pre-messenger RNA, then mature mRNA, and then finally into the polypeptide. Okay, so you should be able to name these strands, and there's the answers there. So, quick recap. First step of transcription is the initiation. All right, so you can see that promoter region, and that's where the RNA polymerase will bind and unzips the DNA strand. Okay, elongation, where free-floating nucleotides get turned into the brand new pre-messenger RNA molecule as the RNA polymerase runs down the strand. And lastly, termination, where the RNA polymerase flings off of the strand at the termination site, obviously stop codons, and now you have your entire pre-messenger RNA created. Awesome. So we're going to look at RNA processing. Really should be a quicker video, this one, because there's not too many steps to this. Awesome. There are your dot points. Let's continue on. So essentially, transcription has occurred to create pre-messenger RNA. So that's our first part of our 3 different stages. Okay, and now we're looking at the RNA processing. Remembering that this only occurs in eukaryotic organisms. So this will not happen at all if this is a prokaryote. Okay. Awesome. So once the pre-messenger RNA is created, it needs to be changed so that it is viable for use in translation. So we have essentially your gene here, okay, and then you have your pre-messenger RNA molecule coded exactly the same. So you have the leader region, okay, exons and introns, and then the terminator region. So it involves the modification of the pre-messenger RNA to make it mature mRNA, okay? And from that, then it gets to the last step, which is the creation of the protein. Awesome. So what's the processes? So the modifications include the removal of the introns. So exons are blue, introns are red here. So you can see all the red parts of the molecule get cut out. Okay. And there's 2 new things here on the mature mRNA compared to the pre-messenger RNA. Okay, so we still have the leader region and the terminator sequence. We still have our exons because they're expressed, but we don't have the introns. And now we have this over here, which you can see is a carbon and 3 hydrogens. All right, so we call that the methyl guanine cap, okay, because it has lots of guanines in it. Okay, so that's on the 5' end. So 5' methyl guanine cap here. All right, and then you have all the adenines on the end, so a poly-A tail, polyadenine tail, because there's lots and lots of them, multiple adenine tail. And that's on the 3' end, hence why it's the tail. Okay, so the tail always goes at the back, so it's at the 3' end. All right, you can think of the methyl G cap. Your cap goes on your head. Okay, so it's at the start. All right, and this is essentially called splicing, where we get our introns cut out. Okay, they must be removed, and the regions corresponding to the other exons must be joined. All right, and this is done by a classic molecule called a spliceosome. All right, so it's like what we call molecular scissors, and it will cut those introns out. All right, leaving just the exons remaining to be joined together through other means, such as non-homologous end joining and other things like that. Okay, it could also be ligase. And then lastly, all right, 5' methyl G cap added to the front, and then the 3' poly-A tail added to the end of the molecule. And that creates our mature messenger RNA. All right, there's another thing called alternate splicing as well. You should just be aware of this. So sometimes exons can also be removed during the splicing process. Okay, so introns are always removed, but occasionally, if this alternative splicing happens, one of these exons might be removed too. Okay, it allows for a single gene to give rise to many different messenger RNA strands. All right, so this could be one gene, but it can code for multiple things. Okay, so if we call back to the proteome when we looked at that, yeah, one gene might code for up to 40 different proteins, and this is one of the reasons why. Okay, alternate splicing. Because as we know, this is going to be the mature mRNA, which then ends up at the ribosome being translated into the protein. Okay, and that these proteins are all going to be different. All right, awesome. So it's just something to be aware of. Cool, and that concludes the video for today. So I hope you enjoyed it. It's a bit of a quicker one, and we'll get on to translation in the next video. Have a nice day. Like, comment, subscribe.